Hello everyone, in this presentation we will be discussing about restriction fragment length polymorphism RFLP. RFLP is a technique that exploits variations in homologous DNA sequences. There are several applications of RFLP. First, it can be used in paternity cases to determine the biological parents of the child. For example, to determine if Jack is the father of Jill's child named Pearl, through the extraction of DNA from white blood cells from all three individuals and subjected to RFLP analysis. On the left diagram, the result shows that Pearl inherited the 12.4 kilobases fragment from Jill and the 12.3 kilobases fragment from Jack. Thus, Jack could be the father. On the other hand, right diagram is an example of negative result which shows Jack is not the father of Pearl, since Pearl has a band of about 6 kilobase and Jack does not have. Besides, RFLP is also used to determine the disease status of an individual, for example, cystic fibrosis and sickle cell anemia. RFLP is also used to measure the recombination rates, which can lead to a genetic map. It can help to measure the genetic distance between two RFLP loci, whether they are a part of genes or not. However, RFLP has some limitations. A large sample size is required to run a RFLP gel. Heavily damaged DNA cannot be used. Contamination of sample may cause amplification other than the desired DNA, and isolation of DNA sample can be a laborious and time-consuming.
Now, let us learn about how the RFLP can detect the sickle cell anemia. First, let us look about the background of the sickle cell anemia. It's a disease whereby the hemoglobin is abnormal, causing the red blood cell to become rigid and curve into a C shape. It results from the homozygous sickle cell allele at the beta globin gene locus. The S allele will differ from the Y type allele by the substitution of the A in the wild type to a T in the second position of a codon in the beta chain gene. So this substitution will change the GAG codon to the GTG codon that result in the replacement of a glutamic acid by the valide. In the wild type allele, the restriction enzyme recognizes the sequence of the CTGAGG that occupies the codon 5, 6, and 7 and will cut between the C and T. So this will produce a 2 fragment of the short DNA. Gel electrophoresis is then carried out to separate the fragment of the DNA according to their molecular weight. Then, a DNA probe is added to allow the hybridization of the DNA fragment. In this case, the probe will bind to the short fragment as shown by the darker red arrow. In the S allele, the sequence of the CTGAGG is lost due to the substitution of A by the T. So the restriction enzyme fails to recognize the restriction site and cannot cut the DNA. Therefore, the DNA probe will bind to the larger fragment, which between the blue arrow. The abnormality of the gene can be detected based on the DNA band formed on the gel. Besides that, RFLP marker can help us to trace the inheritance patterns. The DNA fragment of a family member are also placed directly onto the gel for separation. For example, if both parents are heterozygous for the gene, there's a 1 in 4 chance that child will have a sickle cell anemia.